The CoronaVac vaccine against COVID-19, produced by Chinese pharmaceutical firm Sinovac, has been approved for use in 39 countries around the world, with more than 1 billion doses already distributed. Today, I'll take you through the scientific literature to give you everything you need to know about Sinovac's effectiveness. Specifically, I'll tell you what is known about how effective Sinovac is at preventing infection, hospitalization, and death, how effectiveness changes against variants such as Gamma or Delta, how quickly immunity drops after vaccination, and how Sinovac compares to other vaccines. And as a bonus, we'll look at a study that actually ranks vaccines for their effectiveness. This is All Things Public Health. I'm Anthony Custer. Let's get started. So if you're returning to my channel, you may have seen a previous video where I discussed 7 COVID vaccines that had published phase 3 trial data in peer-reviewed journals. Notably, Sinovac was absent from that video because they hadn't published. Well, in the time since that video went online, interim phase 3 trial data from Sinovac has been published. Before we look at that study though, what kind of vaccine is Sinovac? Is it like other COVID vaccines? Sinovac's CoronaVac vaccine is an inactivated virus. This is the most conventional type of vaccine, using the same technology as the polio vaccine in the 1950s. Essentially, live SARS-CoV-2 virus was collected from infected patients in China at the beginning of the outbreak. Then, the virus was mass-produced in monkey kidney cells, extracted, then treated with chemicals to inactivate it, rendering the virus unable to reproduce. This is different from almost all other COVID vaccines, including the mRNA vaccines from Pfizer and Moderna, as well as the viral vector vaccines from AstraZeneca, Sputnik, and Johnson & Johnson, which all stimulate the in vivo production of viral spike proteins. Only Sinopharm and the Indian Bharat vaccines use the inactivated virus approach that is found in Sinovac. So, does it work? Well, wait, how is vaccine effectiveness actually measured? Vaccine effectiveness cannot be determined from observed trends, like, for example, if infections or hospitalizations drop in a city or country once a vaccine is administered in the population. That trend tells us nothing about the vaccine. This is because there's no way to isolate what factor caused that decline to occur. It may have been the vaccine, or it may have been several other factors unrelated to the vaccine. Instead, a study design needs to be able to measure both an outcome and a factor in each individual. The outcome is usually infection or hospitalization or death, and the factor would be whether that person has had a vaccine. The most reliable way to do this is with phase 3 clinical trials, a type of study called a randomized placebo-controlled trial where participants are randomly assigned into vaccine or placebo groups. However, effectiveness can also be determined outside of trials using real-world data through 2 other study designs: retrospective test-negative case control studies and prospective cohort studies. We'll be looking at data on Sinovac from all 3 of these study designs. So let's get into it. Sinovac had phase 3 trials registered in Brazil, Chile, Indonesia, and Turkey. However, only data out of Turkey was used in the phase 3 trial publication, which came out in The Lancet on July 8th. The analysis included 10,029 participants with 2/3 assigned to the vaccine and 1/3 assigned to a placebo. The results found that the vaccine was 83.5% effective at preventing symptomatic infection among fully vaccinated people. This number places it in the middle of vaccines for effectiveness based on phase 3 trial data alone. Notice though that the 95% confidence interval, this bar, is rather large. This high uncertainty is because the sample size in this phase 3 trial is small. It had only 10,000 participants, compared to 20,000 to 40,000 in most other studies. Also, this effectiveness calculation was based on relatively few observed COVID-19 cases, only 41 in total, with 9 in the vaccine group and 32 in the placebo group. Most other studies made conclusions based on hundreds of observed cases. So, Sinovac reports a relatively high vaccine effectiveness from this phase 3 trial, but it's based on a fairly narrow set of data. For a clear answer, we need to look at studies with more data. A much better study comes out of Chile. It's a cohort study, which means it looked at an entire population of people. In this case, every adult in the National Public Health Insurance Program of Chile, which ultimately included data on 10.2 million people covering 3 months from February 2nd to May 1st, 2021. In that time, more than 4 million people received the Sinovac vaccine. And overall, more than 218,000 people in the cohort got COVID-19, resulting in over 22,000 hospitalizations and 4,000 deaths. So with this much richer data set, what did they find? Among people fully vaccinated with Sinovac, they estimate the vaccine was 65.9% effective for preventing COVID-19, 87.5% effective for preventing hospitalization, and 86.3% effective for preventing death from COVID. So, with a much larger data set, we see that Sinovac's actual vaccine effectiveness is probably about 66% for preventing symptomatic illness, a much lower number than the 83% reported from its published phase 3 trial. But could any of those differences be due to differences in variants? Other vaccines, including AstraZeneca and Pfizer, have reduced effectiveness among the variants Alpha, Beta, Gamma, and Delta. However, no such study has directly compared the effectiveness of Sinovac among different variants as of August. What we can do, however, is look at genomic sequencing data to see what variants were spreading throughout each study. For example, the phase 3 study in Turkey covered the period from September 2020 to March 2021. Looking at genomic sequencing data out of Turkey, we see that D614G was circulating. This form of the virus predates Alpha, Beta, and all other variants and is close to the original wild-type virus out of Wuhan. On the other hand, the cohort study from Chile took place from February to May 2021. There, Lambda and Gamma were widely circulating. Using this information, we can see that variants may partly explain differences in the effectiveness estimates seen between Turkey and Chile. And one more study out of Brazil shows how Sinovac did against Gamma. In the study, which looked at the elderly in Sao Paulo, effectiveness of Sinovac against Gamma was 46.8% for infection, 55.5% for hospitalization, and 61.2% against death. What we see is that Sinovac's effectiveness against all outcomes drops with older age and with more virus mutations. But what about Delta? Researchers in Thailand collected blood samples from healthcare workers who had been fully vaccinated with Sinovac and then tested to see if antibodies in that blood could neutralize the virus, looking at the original viral strain known as wild type and also at the variants Alpha, Beta, and Delta. What they found was that antibodies from people vaccinated with Sinovac were very good at neutralizing wild type SARS-CoV-2, able to do it about 98% of the time. This makes sense since Sinovac is just an inactivated form of the wild type virus. However, against Alpha and Beta, that neutralization rate dropped to 75 and 70%. Against Delta, Sinovac antibodies could only neutralize the virus 48% of the time. So, Sinovac's effectiveness is reduced against the current variants. But what about waning immunity in our bodies over time? We know already that immunity requires time to develop. Even after receiving a full vaccine dose, the full immune response generally develops about 2 weeks later. But what about after that? Once immunity does develop, how long does it last? Researchers in both Brazil and Chile have shown that Sinovac's antibody levels decline over time. In Brazil, researchers looked at 133 healthcare workers that received 2 full doses of Sinovac and collected blood samples from them starting on the day of the first dose, then again at days 10, 20, 40, 60, and 110 days after the first dose. Then, they measured levels of antibodies against SARS-CoV-2 proteins. After the first dose, virtually no antibodies developed. Then, about 2 weeks after the second dose, so 40 days after the first one, antibodies peaked. Then, they steadily dropped. Antibody levels dropped in half about every 1 to 2 months. A more comprehensive study from Chile found the same thing. Antibody testing showed declining seropositivity among CoronaVac recipients, declining to about 50% after 20 weeks. Over the same time period, seropositivity remained at nearly 95% among recipients of the vaccine from Pfizer. Now, the immune system has other tools beyond neutralizing antibodies, so this doesn't mean a person's immune system is completely helpless 6 months after vaccination with Sinovac. But a major study in the journal Nature shows that neutralizing antibody levels are highly predictive of immune protection. So with that in mind, let's rank the vaccines. Looking strictly at phase 3 trial data, Sinovac might appear to be somewhere in the middle of the pack among COVID vaccines. However, If you know my vaccine effectiveness video, you know that a number of factors confound this comparison. To get around these confounding factors, this study from the journal Nature attempts to make an apples-to-apples comparison of vaccine effectiveness and ranks the vaccines using antibody neutralization levels. So, who's number one? According to this study, Moderna is the most effective COVID vaccine. Next is Novavax, third is Pfizer, and fourth is Sputnik. Then we start to drop down to the next tier of vaccines. At 5, we have AstraZeneca, behind that is Johnson & Johnson, and coming in last is Sinovac. So what does all this mean? Because Sinovac has the lowest levels of neutralizing antibodies among widely used COVID vaccines, breakthrough infections may be more likely with Sinovac compared to other vaccines. Booster shots may be needed earlier for Sinovac compared to other vaccines as well. Many countries, such as Thailand, have already decided to give a third shot of Pfizer to boost healthcare workers previously vaccinated with Sinovac. Sinovac is still needed and useful. Vaccination with Sinovac is still safer than no vaccine. In fact, several countries will rely on Sinovac since many other vaccines are logistically unfeasible for many parts of the world. For example, mRNA vaccines require extremely cold temperatures of -70 Celsius. The cold chain infrastructure to maintain this temperature all the way to the injection point doesn't exist in many parts of the world. Additionally, certain populations may benefit from an inactivated virus vaccine as opposed to other technologies. Altogether, it's good to have a variety of vaccine choices in the world, as long as we understand the differences between them. That's it for today. Thanks for watching. Please consider subscribing and be sure to check out one of these other videos while you're here.